I want to begin our first session today. So our first session is entitled, it's up there, What Does Sex and Gender Have to Do with It? I'm trying not to be Tina Turner up here. And how, these speakers were chosen for very good reasons, which you'll see as the talks go on, about how studying sex and gender-based analysis can enhance our knowledge on mental health for all of us. And up first is Dr. Robert Paul Juster from the Université de Montréal. I've known Robert Paul for a long time, and I know you're in for a treat. So please welcome.
Good morning, everyone. Can you hear me well? Excellent. Thank you, Lisa, for the invitation. I'm an integrative neuroscientist who's very interested in how the social environment shapes biology. I use sex and gender approaches to identify stress physiology in relation to mental health. And in this presentation, I'm really going to show you different ways that we can look at sex as a biological variable, as well as sociocultural gender as a sociocultural variable, and also looking at sexual orientation. I'm very interested in looking at the LGBTQ+ community as a model of a group that's exposed to more stigma, stress, and strain, but also remarkable resilience. So I'm a stress scientist. I was trained by Sonia Lupien, who's the queen of stress in Quebec, who taught us that stress, as defined by her own mentor, Bruce McEwen, who was also Lisa's mentor, that stress is broadly defined as a real or interpreted threat, the physiological or psychological integrity of an individual that results in biological and behavioral responses. So in blue at the bottom, you have the sympathetic adrenal medullary axis, which leads to the release of catecholamines. You feel this right away within seconds, your blood pressure increases. And then at the top, you have the green line, which is the hypothalamic pituitary adrenal axis, and this leads to the release of cortisol that travels around our bodies and transforms fat into sugar, and essentially gives us the energy required to maintain the fight or flight response. It says something, unmute to speak to start. Do I need to do? All right. I've spent the better part of my, no stress, no stress.
There we go.
All right. All right. So I've spent the better part of my life stressing other people out. And what we do is we expose them to a stressor called the Trier social stress test, which involves a mental arithmetic and a public speaking task. So a sort of challenge-based experience. And right off the bat, I'll show you that men in general seem to react more than women. But women will actually report on questionnaires being more stressed. So right off the bat, we have this potential sex and gender difference going on. We also like to look at cortisol over the course of the day. So we have a rise when we wake up called the cortisol awakening response, and then levels that are at their lowest right before going to bed. And there's a lot of psychiatric conditions that have particularities with this profile. So depression seems to be related to this hyperproduction of cortisol and very high levels before going to bed, while things like burnout, fatigue, discrimination are related to actually this blunting of the diurnal rhythm. So any physiological change in our homeostasis as we're adjusting to our environment is what's called allostasis. It's the stability through change of how our bodies and our brains adapt to our environment. And it's a concept that was taken up in the stress world in order to identify the costs that our body and our brain pays when chronically stressed. So allostasis is the idea that to maintain stability, an organism must vary all parameters of its internal milieu and match them. Do I only have 14 minutes left? Okay, so it's really stressful. So we want to have a stress response. It's adaptive. It's what allowed us to survive as a species. We want to have a stress response if we're Bambi in the forest. We also want to have a good memory of where that bear is. So the systems that regulate the stress response are also regions that are involved in cognition. And there are different profiles. So repeated activation of the HP axis that leads to dysregulation, lack of adaptation. So people with low self-esteem have a harder time adjusting to stressful situations over time. Prolonged response. This is unfortunately something that happens in aging. The HP axis gets a little bit worn out. And finally, an inadequate response where we don't produce enough cortisol. As I mentioned, this is something we see in fatigue and burnout, in trauma, and in a lot of the discrimination literature. Now, this is an image generated by AI of allostatic load. So if anyone's seen the movie Oppenheimer, it's pretty much allostatic load in a nutshell. Allostatic load is essentially the cost that our body and our brain pays for being chronically stressed. It's the wear and tear of chronic stress in synergy with unhealthy behaviors. You can think of it a bit as like the camel that breaks the straw that breaks the camel's back. And it involves more than just cortisol. It's the central nervous system, it's metabolism, it's cardiovascular functioning, and it's immune functioning. And essentially, allostatic load is a measure of dysregulated biomarkers that for an individual. There is a disease time course, so people believe that first it's neuroendocrine and metabolic stress and dysregulation in all of these stress hormones that leads to cellular and subcellular changes. This is some work of Martin Picard at Columbia, looking at mitochondria and telomeres. And this ultimately over time is going to lead to cellular aging, accelerated aging, and dysregulation of what's called secondary outcomes, which is essentially all your traditional metabolic and cardiovascular biomarkers. Being stressed costs energy. So there is also this idea that allostasis demands energy, but it's adaptive, it's healthy, but then becomes allostatic states and allostatic load. And finally, allostatic overload is when the systems just give up, and it's a total breakdown of all these systems. This is more than just a theory. It's something we can actually measure. And in first studies that looked at it were among successful agers. And you see here 10 different biomarkers that are all in some way related to stress physiology. And the advantage of this allostatic load index is you're not just looking at one biomarker at a time, you're looking at an ensemble. And you're not just looking at values that pass a clinical threshold, you're looking at subclinical thresholds as well. So it's a very comprehensive way of understanding the physiology of the individuals. It also, in my opinion, and a lot of my colleagues, has potential applications in clinics. My dream is that one day when you get your results for your blood draw, there wouldn't just be whether you're high or low, there'd be an allostatic load column that would inform clinicians of potential disease or multimorbidity. And in the context of psychiatry, this could be extremely important. So allostatic load was validated in a lot of studies around the world, and really the factor that's the most important is aging. Unfortunately, the older we get, the higher our allostatic load, non-white race ethnicity, low social economic status, and finally sex. And this is where I started my career with Sonia. There were no clear sex differences in the literature. So I wanted to first do a review to identify what are some of the causes. So these are all the multicolored triangles down here. And these are all the different outcomes of allostatic load. So we're talking of physical health problems, cognitive health problems, and psychiatric problems. So I've dedicated my career to understanding what are the factors related to sex and gender that are related to allostatic load and to cortisol. And these are sort of an ensemble of all the different factors that I've looked at. Today's presentation is going to focus mostly on sexual orientation and gender identity. That actually informs a lot of my thinking about gender inequalities and structural sexism that I'll talk about at the end. So first, with regards to sex assigned at birth, I mentioned at the beginning that men seem to respond more to stress responses involving challenge, but women seem to be more responsive to stressors involving social rejection. So right off the bat, the nature of the stressor might be gendered. Here's a really interesting study. So this shows you cortisol is higher among men overall in a stress task. Now, if the men come with their wives, they show a big benefit in terms of less cortisol, but the women actually show much higher levels. Now, when women come with and are with a same-sex stranger, they show a decrease in cortisol, but now the dudes are all stressed out. So interesting social support is this showing this sort of gendered effect on physiology. Now for allostatic load, this was a paper that Lisa was, I believe, the guest editor. We wanted to see allostatic load, and we found that actually men seem to have a higher allostatic load than women in studies where it's reported. However, when you start nuancing some of the associations among women, they seem to be very gendered. So women that attend religious services more often have lower allostatic load. Women that are single mothers have higher allostatic load. Women that experience more job stress have higher allostatic load. Women with hostile personality traits and masculinity have higher allostatic load. And finally, this is another interesting finding. In Taiwan, older men benefit a lot more by being married to wives, but wives benefit a lot more by having good bonds with their same-sex friends. To mirror this is a study that just came out in my lab looking at the health and retirement study, where we found that allostatic load among older adults that have just retired is actually correlated. But over 4 years, it's the husband's allostatic load that predicts the wife's allostatic load, but not the other way around. So, sorry. Now for gonadal hormones, I mentioned that cortisol is higher among men for these challenge stressors. Oral contraception lowers cortisol reactivity, and the menstrual cycle is related, so women in the luteal phase have higher cortisol. No differences in diurnal cortisol based on this. All of these studies assume a certain profile of hormones, but don't actually measure it. So in one of my doctoral studies, we measured testosterone, estradiol, and progesterone for both men and women and looked at how it would relate to cortisol. What you see here is what we normally see in the literature. So A1, higher cortisol reactivity among men. This is standard in the literature, and then no differences in diurnal cortisol, B1. But when we actually adjust for individual differences in hormones, in these gonadal hormones, we lose this reactive effect, but we unmask a diurnal difference where women have higher levels of cortisol in the morning. And this is actually being driven by women that have children at home. This is important that biological sex and gonadal hormones is a way of actually looking at sex along continuums rather than just relying on binary sex. Now for sexual orientation, we know that the LGBTQ+ community experiences more stigma and stress. This comes from distal stress processes, so the environment, the violence, as well as internalized processes, so what we call proximal stress processes. And this literature is informed by Mayer's sexual minority stress model that basically says being a minority is related to unique experiences of stress that are in addition to the stress that everyone else experiences, and that increases risk of mental health disorders. And of course, there's also coping and resilience differences unique to the communities as well. This model was taken up for gender identity as well, and it's exactly the same idea. Unique forms of stressors, increased risk of mental health, physical health, as well as unique resilience factors. Very little research had actually looked at stress biomarkers, and I had the fortune of doing a study early on in my career in Canada, where we looked at 87 individuals, half of which were LGB, half of which were heterosexual. This was our protocol. It's very typical of all the data that I'm showing you today. People come in for a first visit where we do a blood draw, they go home and they do some sampling, and then they come back for a second visit where we stress them out halfway through. And we also measured sex hormones before and after the stressor. And what we found is that lesbian and bisexual women in the panel A had higher levels of testosterone, no differences for men, no differences for estradiol for either men or women for bisexual orientation, and then progesterone higher in lesbian and bisexual women. Now, a lot of literature in the 80s in psychiatry looked at sex hormones in relation to sexual orientation, but this literature was very flawed because it never actually took into account psychosocial experiences. And so when we actually looked at what were daily stressors in the lives of these women, that was actually what was driving their higher levels of testosterone and progesterone. It was also predicting higher reactivity in terms of cortisol. So you see that lesbian and bisexual women are showing higher levels of cortisol 40 minutes after the stressor compared to heterosexual women. For the men, we found actually the complete opposite. Heterosexual men showed much higher levels than gay and bisexual men. I'm not going to focus too much on the men today because it's women mind. But if you look at the gay and bisexual men here, they have higher heart rate. So it's interesting, just for cortisol, it's actually lower, but for heart rate, it's actually higher. No differences among the women. Now, for diurnal cortisol, we didn't find any differences based on sexual orientation, but I had the good idea of asking people about their coming out process. And what I found was that those that had completely come out of the closet to their family and their friends had lower levels of cortisol in the morning. No differences in mental health. Even kind of surprising, gay and bisexual men had less depression than heterosexual men. This does not correspond with sexual minority theory. And then actually the lesbian and bisexual women had a trend towards higher depression, but it was actually being driven by by individuals that had come out of the closet. So those that had completely come out had less anxiety, less depression, and less burnout. Now for allostatic load, we had 20 different biomarkers, and we found that gay and bisexual, I have 5 minutes, right? This is the thing on? All right. Excellent. Gay and bisexual men had lower allostatic load than heterosexual men. I'm not going to talk about that. I'm going to talk about the fact that there wasn't any difference among the women. And that's something that's going to repeat itself in my studies, and I'm going to talk about why I think that is. We didn't find any differences on coming out, but secondary data analysis found that those that engaged in less avoidance during their coming out process had lower allostatic load. So this really shows the importance of the coming out process. But of course, the experience of Canada is different than it is in Chechnya. So I don't want to assume that just coming out of the closet is healthy for everyone. It's very space specific. There's some limitations to this study. I think we have a selection bias in terms of who's coming. We also got some good fortune that Steve Colbert picked up our study and said that it would need to be replicated with real humans, so not Canadians. So that was the inspiration for my postdoctoral work to go to the US and see if they are as crazy as we are. They are. And again, I found that gay men had lower allostatic load than heterosexual men, but this time because we had enough power, we had 14,000 people. Bisexual men were the ones with the highest allostatic load. Now, no differences in sexual orientation for women. And this was surprising because the lesbian women actually had higher BMI, but their lipid profiles were actually healthy compared to heterosexual women. So I'm not exactly sure what could be driving this. Future work should actually look at health behaviors. This is me during my grant writing season. It makes sense that allostatic load would be correlated with health behaviors. So I think that's something that needs to be looked at further in this literature. This is a review of 43 studies using the database that I worked with, the National Health and Nutrition Examination Survey. And if you see a profile that's dark blue, that means the profile is more problematic. And if it's green, it means it's healthier. And so you can see for the gay men, there's a lot of kind of mixed results. Same thing for the lesbian women. But then for bisexual women as well as bisexual men, it's really the worst profile. And that's consistent with a lot of the psychology literature. One final study that I did during my postdoc with Mark Hatzenbuehler at Harvard looked at structural stigma. So the laws in place in different states that protect sexual minorities. And we were able to index that and look at how it related to allostatic load. This is what it looks like on average between 2001 to 2014. And what we found was that gay and bisexual men that live in states with more protections have lower allostatic load, but we didn't find the result in women. And I suspect the reason why is that if we were to actually take measures of structural sexism, we would have a completely different profile. And I'm going to talk about that a little bit at the end of my presentation. Now for gender identity, I just want to point out that it's incredibly important to engage in participatory research with the community. This was something we did during the pandemic to really ask the community what kind of research they wanted. And I partnered with Zachary Dubois, who's really been a leader of biomarker integration among trans health. And this is a study where we found that transgender men that report more stress with their transition, more stress with coming out, and more stress with public restrooms have higher cortisol levels over the course of the day. In the states of Massachusetts and Vermont, we found that those individuals that lived in, even in these progressive states, lived in less progressive spaces, so more conservative spaces, these were the ones that had higher allostatic load. Everything that was related to transition stress was related much more to mental health. So we're following up with some work. This was done before and after the last US election. It's the SPC study, sociopolitical context. And these are just some of the papers that have come out. And one of the papers that just came out found that enacted stigma was related to a blunted profile of cortisol, something that we see in trauma and fatigue literature. But greater connectedness to the community was actually related to a healthier and more dynamic diurnal rhythm. The approach that we took of looking at structural stigma for sexual minorities could also be done for transgender rights in the US, but also in Canada. We're in a period right now where there's a lot of legislation coming out. So I think this kind of work can still be done here. And my work right now in my lab is trying to add to sexual minority stress model by integrating biomarkers and better understanding resilience, so coping strategies, social support, and community resources. And I was fortunate to have a CIHR sex and gender chair where we're doing this work right now. And in closing, I just want to make parallels to gender norms and institutionalized gender, because I think a lot of the work that, a lot of the things that are going on in the US right now are applied. So this is a study that found that women that live in states with reproductive bans have higher levels of depression. And just in closing, I'd point out the work of Patricia Homan that looks at structural stigma, structural sexism across the US in relation to health outcomes. And I would think it would be really interesting to take this approach of structural sexism to look at biomarkers like allostatic load. And I think in an intersectional perspective, if we were to have measures of structural stigma related to sexual orientation, to gender identity, to race ethnicity, to sexism, that could be a way of actually looking at intersectionality at a macroscopic level. So in closing, it takes a village and even a gay village to do all this research. I want to thank my growing lab and also encourage you to add us on your socials because we need more followers. We do our things in both French and English, so don't be scared if it's all very French at first. Thank you very much.
I'm very happy to say that we have time for a couple of questions.
I have been, for those on the stream, it's Megan talking, and I was a speaker yesterday on women and autism. Thank you so much for your presentation. We talked a little bit last night, and I mean, wow. I think I speak for everybody in here. Like, that was neat, the amount of information. Two things I wanted to ask, two things I wanted to comment on. First and foremost, thank you for asking that community about what they want the research to be on. Too often an assumption is made by researchers as to what needs to be done. And it's asking those community members, when we talked about working with community, interdisciplinary work, that is something you can do. And I think that is well done and deserves a round of applause.
Oh, thank you.
Now, getting to my question, I have so many ideas for your research going forward, but I'm going to save that. Okay. One thing before I ask my question, because perhaps I've misunderstood, the studies in the beginning that you referenced in terms of couples, where we all thought it was quite humorous, and I can say my husband for sure, there's some stress there at times when he's around. Were those the additional follow-up information on individuals that were gay, lesbian, etc.? Was that referencing individuals only, or as in couples as well?
That's a great question. It was couples.
It was couples. So that was my question for you then. Yeah. So see, you've answered it for me, is what did the allostatic load look like in same-sex couples as well as in, you know, in non-binary couples? Was there any difference in that? So that was my question, but I thought, hang on a minute, maybe I misunderstood with the individual. I just want
to make sure that I got the right study. This was the one, there's a lot of slides.
There's a lot of slides, but it was so interactive. And
here we go. This one?
I believe so, yes.
So this was with heterosexual couples. This sample actually does have an LGBT subsample. But we unfortunately didn't include them because we didn't have enough power, which sucks. That being said, this study here where we're looking at the stress reactivity profiles, this one.
Yes, that's
this was done with hetero, like I don't even think they measured people's sexual orientation. I think it's just assuming that they're heterosexual and cisgender. But I'm proud to say that we are actually conducting a study right now where we're bringing in LGBT couples as well as heterosexual cisgender couples. And we're stressing one of the partners out and seeing how the other partner interacts and to see whether we will see the same thing as a function of sexual orientation and gender identity.
So to be announced. Thank you.
Yes, one quick question. Oh, you have, use a microphone, because the online community can't hear if you don't use a microphone. But yeah, masterclass.
Oh, thank
And
you.
Just
I had a lot more.
Just a quick question. What do you use to stress them out?
So it's the triage social stress test. So they come into the lab, they know, now we've actually put it in our consent forms and even in our advertisement because we don't want to deceive them. We used to deceive them, but we don't do that anymore. What was I saying? Yeah, so they come in and then we give them instructions. We say, you're going to be doing this task. We want you to prepare for a mock job interview. We give them 10 minutes to prepare. And then they go in a room where they're alone, and our team is on the other side of a one-way thing. You can also do it face-to-face, but that's actually more stressful to people who are the judges. So that's the task that we use. Yeah.
I am very sorry, Robert Paul, that I pushed you off the stage because you deserve the stage, but we now make room for Dr. Georgia Hodes from Virginia Tech, who will take us into probably the animal kingdom a little bit.
Yep.
Take it away, Georgia.
I'm a good segue since I'm going to talk about both humans and animals. First of all, I want to thank Lisa and CAMH for having me here. It's such a pleasure to get to talk to you all, and it's just been a wonderful meeting so far. So when I was starting my own lab, I was really interested in this idea of how does the immune system contribute to mood disorders? And I was particularly interested in what's happening in the periphery. So what's happening in your body that may end up influencing your brain, and particularly things like the cytokines you produce, which you can think of as immunotransmitters, right? In the same way we think of this as neurotransmitters. They communicate with other types of cells in your body, but also what's happening in the brain. And so I'm going to talk a little bit about both sides of this today. And the study that had really gotten me excited about the idea of looking at sex differences in major depressive disorder was a study by Michael Mays from the 90s. And one of the things he had shown was that people who have depression have more white blood cells than people who don't. And I had seen this in an animal model as well. But when we looked at men, they had more monocytes, so more innate immune cells. This is the first line of defense. Whereas when we looked at women, they actually had more lymphocytes, which is part of the adaptive immune system. This is what we think of as immunological memory. And so I was excited by this idea that there may be different mechanisms involved in how men and women experience immune changes during depression. And this paper had come out, which showed that men did have really a weaker response of their immune system to a variety of illnesses. So what they were basically saying is that man flu is real. That said, it's, I would point out it's all male authors, so there might be some bias. But we know that women are more responsive to vaccines, that they actually have more T cells than men. They're also more likely to develop autoimmune diseases. And when we think about the immune system, right, it's a double-edged sword, that you want a strong immune system that's going to protect you, but because it's a strong immune system, it may also protect you from things you don't necessarily need to be protected from. It may think things are more dangerous to you than they actually are. And you also have this greater likelihood of developing autoimmune diseases. Most autoimmune diseases are at least twice as likely to occur in women than as men. And we see that with depression as well, right? That it's 2 or 3 times more likely to occur in women than men, particularly when you're talking about atypical or somatic depression, which involves pain. So when I started out, I wanted to know, are men and women having the same immune response to major depressive disorder? And I figured, I'm doing mouse work. I figured this is going to be really easy. I'm going to go through the literature and I'm going to pick out the cytokines that are altered in women and altered in men and compare them. And then I went through the literature. And first of all, nothing was segregated by sex. And it was really hard to find out the sex of the individuals. Also, they combined men and women from age 18 to 80. And we know all of these immune cells have estrogen and androgen receptors. So there's a lot of issues with what's been in the literature. So I said, okay, I have friends who collect human samples. I was part of building a blood bank at Mount Sinai. I'll ask them for samples. And so I wanted samples from women who had not yet gone through menopause. And that was also quite difficult to get. So I do have to point out that the ends are relatively low in this study, but I was able to get about 78 people. They divided into people who were controls, who were having they were going to a doctor for the first time for an episode of depression. So I can't say it's their first episode of depression, but it's the first time they're trying to get treated, versus people who were treatment resistant, meaning that they had failed at least 2 treatments. And these are people who are within a relatively typical BMI. They were sort of medium depressed in terms of their quick inventory of depressive symptomology score. And their age on average was about 34. We used 55 as a cutoff. And so we took their blood and we looked at their cytokines. We used a multiplex panel, which allows me to look at, about 30 cytokines in the same individual's blood at the same time. And, we didn't really see that much when we just looked at healthy controls versus major depressive disorder. We saw that, VEGF, interferon gamma, interleukin 17A were elevated in people who had depression. We broke it down by treatment resistance. This didn't really change other than the fact that it's mostly driven by people with treatment resistance. And then we segregated by sex. And what we found was that there were no changes in men whatsoever, and that all of the changes were in treatment resistant women. And this is just, there were far more changes, right? We were able to actually see how large, this is all normalized values. Some of these were, 5 times as high in these women with treatment resistant depression. And just to sort of bring in that point more, that little pink dot on the end, that's IL-15, that's women who are having their first episode of depression. Everything else that you saw that was altered was being driven by these women with treatment resistant depression. And so I think, when we think about these sets of data, one of the reasons that we don't see as many changes at baseline in cytokines may be because we're combining males and females, as well as people who are treatment resistant versus having their first episode. And so we really need to sort of pull this together and start understanding it better. So I do work in mice. The model I work with is a variable stress model. So we use a combination of repeated foot shock, tail suspension, and restraint tube. We then give them a behavioral assay. We can do a bunch of different behaviors over different days to try and better match up with what we see when we're testing humans with depression, where we're looking across different types of symptoms. In this case, we create a Z-score that's a stress susceptibility score. And I'm going to be really talking about the 6-day of stress time point. And the reason I'm interested in that particular time point is because if you take these males and females and you stress them for 6 days, and then you run their behavioral profiles, across behaviors, females are more stress susceptible than males. That we get behavioral effects in the females that we do not see in the males. If we go out to 28 days, then we see effects in both sexes. And I'll talk a little bit more about how the mechanisms behind those differ. I was lucky enough to work with Marianne Sine, and she works with 4-core genotype mice. I'm not going to go into a lot of detail about these mice because she's going to talk about them later. But just to say that one of the things that's so amazing about these mice is that you can actually take apart what's based on gonadal hormones versus genetic sex, right? So you can dissociate gonadal hormones from genetic sex. So you can have XY mice that have ovaries, and you have XX mice that have testes. And so in collaboration with Marianne, we looked at this effect of stress, and we found that this was really driven by the chromosomes, the sex chromosomes, that it didn't matter whether the mice had ovaries or testes. If they were XX, they were stress responsive after 6 days, whereas if they were XY, they were not, even when they had ovaries. We also looked at their blood, and again, we see, so first of all, I want to point out, we see far fewer cytokines altered in the blood of animals who are stressed, right? If you learn anything today, stress is not depression, but it does give us some insight perhaps into how depression starts, right? And I really think this is important, that when we think about things like depression, particularly something like treatment-resistant depression, we're talking about people who have had multiple episodes. And each time they have an episode, particularly if they have an immune response, it's becoming more and more dysregulated. And that's what we're really seeing in these women with treatment-resistant depression, is that we see cytokines that aren't supposed to be all up at the same time being elevated, that they're supposed to balance each other out. And we don't see that anymore in these women. And I think it's really this experience of having multiple episodes. And so this is something I'm really interested in looking at in animals. We did see a couple of the cytokines that matched up between humans and in mice. These were all chemokines. So what they're doing is they're calling different types of immune cells over to an area, right? So in this case, into the blood. And so this is an area that we're still trying to develop. Why do we constantly see these signals that are sort of calling different types of immune cells there, and what does it mean? I'd also like to point out, none of these are the usual candidates that we see in terms of interleukin-6, TNF-alpha, that these are different cytokines, and they're involved actually heavily with the adaptive immune system, and also Th2, right? So what we think of as an anti-inflammatory response rather than an inflammatory response. And so whenever I talk about this research, people say, well, that's really cool. What's happening in the brain? And so I did turn around and start studying what's happening in the brain. How do we go from having a brain like this to a brain like this? And this, my training had been as a neuroscientist, not as an immunologist. And when I was doing my postdoc, I had the fortune to work with Benoit Labonte and Eric Nessler's lab on a study where we actually were able to look at people who had human depression and do sequencing on a variety of areas of their brain. I'm going to be talking about the nucleus accumbens, but one of the things that we found was that there is almost no overlap in the transcriptional signatures of major depressive disorder in human males versus human females. There are very, very few genes that overlap. And this followed a pattern that I had seen in my mice when they had been exposed to either 6 or 28 days of variable stress, that we really don't see a lot of overlap. This, though, is done by looking at how males that are stressed compare to male controls, and same thing for humans, versus females who are stressed versus female controls, right? And seeing what overlaps. So I wanted to do a direct comparison. And when I did that, what I found was that the type of genes that were most regulated were these immune genes, right? And this is immune genes in the nucleus accumbens, in the brain. And so to think about, well, what immune genes should we be looking at, we move to microglia, because these are the innate immune cells of the brain. And so what I'm showing you here are the changes in morphology that occur in microglia. These are incredibly dynamic cells. They are normally in this surveilling state. I like to do my microglia dance. They put out their processes and feel around and see what's going on and try to protect you. And then if there's something there that's not supposed to be there, or something they consider dangerous, they start to pull in those processes and change their morphology so they can move more easily. The problem with our field is that we talk a lot about activated microglia when we're measuring morphology, And what by activated, we mean they're producing cytokines, they're changing their transcription, they're engaging in phagocytosis, but all of these stages are activated, right? And microglia can go back and forth between these stages. So we really don't have a good understanding of what the relationship is between the dynamics of morphology and activation. And I was really interested in this. And I was particularly interested in this in females because almost all of the literature in microglia, particularly in rodents, has been done on males. And in fact, almost all of the findings they have in terms of the importance of microglia are in males. And so I wanted to look in this brain area, the nucleus accumbens, that's involved in reward and emotional processing. And I had a great graduate student who did this. She did a ton of work on this, Masha Seglovia. And she looked at all of these different morphologies. She first went through and scored what they looked like grouped them into these different groups of surveilling, prime, reactive, and amoeboid. And what I'm showing you here is that in the female nucleus accumbens, you actually only see amoeboid microglia after 6 or 28 days of stress in the nucleus accumbens of females. We don't see this in males. Males don't seem to have a responsive nucleus accumbens. We looked at this in another way where we traced these cells and then looked at their area and their number of their branch lengths, and they're pulling in their processes, right? So they're becoming more amoeboid. We've also looked at a phagocytotic marker, CD68, and she was able to show that 6 days of stress increased this marker in the microglia, meaning they're eating more, right? They're going around and eating more. And again, so this is showing that not only are we having a change in morphology, but we're having a change in activation. And so what I really wanted to know is what does this mean? What is this change in activation doing? Is this protective? Is this detrimental? And so I wanted to get, how do I change that activation state, but only in one brain region? And, you know, normally if I was working with neurons, I just inject a virus and a transgene and I could change that one area. But you can't do that with microglia because they're incredibly difficult to transfect. You get a really low level of transfection. So instead, I decided to take a ligand for a receptor that's only on microglia in the brain. So the receptor is CSF1R, and people have used drugs that target CSF1R to try and knock down microglia. So if you get rid of the if you inhibit the receptor, then you wipe out 99.8% of the microglia in the brain. But I didn't want to wipe it out everywhere, and that also wipes out your monocytes as well in the periphery, about 60% of them. So I just wanted to target this receptor. It has two ligands. One of them is interleukin 34, and one of them is CSF1. CSF1 is mostly expressed in the cerebellum in adult animals, whereas IL-34 is expressed really highly in the ventral striatum, and we don't know very much about what it is or what it does. So most of the studies in this have been on CSF1. When I had been at Mount Sinai working with Miriam Murad and Scott Russo, I had looked at IL-34 and I had been interested in it because transcription was decreased in postmortem tissue of the nucleus accumbens of people who had major depressive disorder. And this is in mixed both males and females. When we exposed our female mice to 6 days of stress, we also saw this downregulation of transcription for interleukin 34, so for this ligand to the receptor. So I am very lucky to work with Tim Jerome at Virginia Tech. He's extremely talented molecular biologist and neuroscientist, and he made me a CRISPR for IL-34. So we injected this into the nucleus accumbens of female mice, and we're able to knock it down about 50%, which is exactly what we're seeing with stress. The first thing I'll point out to you is that this is staining for IBA1, which are the microglia in red, and in green is the CD68. And what I'm showing you here is that we're mimicking these effects of stress, that we're getting a decrease in the microglia area and their branch length, and an increase in CD68, this phagocytic marker that you can see very obviously in green in these microglia. And what made me really excited is that we took these mice. So these mice have not been stressed. All we have done is we've taken these mice and we've knocked down IL-34 only in their nucleus accumbens. And we tested them in one of our behaviors. This is a novelty-induced hypophagia. We train our mice to eat Nutella. They like to eat Nutella. We put them into a scary environment, so it's anxiety-provoking. The Nutella is right in the middle of an open field and they have to go out there and get it. And what we saw is it took longer. So just like stressed mice, it actually impacted their behavior, even though these animals were not stressed. And so we saw this when they were in the novel area. So when they were in an anxiety-provoking area, it did not reach significance in the home cage. So it doesn't suggest that it's anhedonia. It suggests it's something about the anxiety of the environment, and it doesn't change their weight. So it's not that they're just not eating. So what I hope I can tell you today is when we talk about the periphery, the main thing I will say is that stress does not equal depression, but we can see overlaps in the immune response, that we definitely see overlaps for chemokines with depression, that there's a possibility, because this is anti-inflammatory, this may be protective, and that most altered cytokines are in treatment-resistant women. When we look at the brain, what I can tell you is that stress impacts microglia morphology in the NAC in a very sex-specific manner that impacts females. That we think IL-34 may be a mechanism by which stress impacts microglia and behavior in the NAC. And I would really like to bring home, which I know I'm preaching to the choir, that sex as a biological variable is really important when we start to think about biomarkers and novel therapies for mood disorders, because what works in males may not work in females. So I just want to finish by closing and thanking the people who worked on this. Jennifer Rainville was my postdoc who did a lot of the blood work. Masha Segovia started all of the microglia work. Dawson Kropp's my current graduate student who's been working on this. And I particularly want to thank Marianne Sine, because several years ago, she came over to me at a meeting and she's like, we're working on the same stuff. Let's work together. And it was, I mean, she's been just the most amazing collaborator. It's been so much fun working with her, but she's so talented and wonderful. And I just so appreciate that that was her response. So thank you guys all for your attention.
I think we have time for 2 questions. I'm looking at the boss over here. You might think I'm the boss. I am not.
You got one over there.
Hi, that was really amazing information. Great talk. Thank you. With the IL-34, did you find that it's associated with a certain reactivity stage of the microglia?
Yeah, so it seems like they're moving into that sort of primed state where they're getting a thickening of processes. They're not necessarily fully pulling them in, but we can see that they're absolutely like thicker and shorter processes.
And so that's more of the pro-inflammatory?
Yeah, well, we know it can be either. So this is the thing with microglia. You can't tell whether it's pro or anti-inflammatory. And anti-inflammatory is a misnomer, right? Anti-inflammatory just means it's there to kill parasitic worms, right? I mean, it does provide also growth factors and things like that, but either form of them can be phagocytic.
Okay. Yeah, thank you.
I love that. Worms. Any other questions? Jennifer, thank you. Jennifer Raven.
Thank you. That was amazing. Can you just talk a little bit more about how we might think about treatments in females versus males?
Absolutely. So, I mean, I think one of the things, especially when we think about these cytokines, right? One of the things Marianne and I have been working on together is to really try and understand how groups of cytokines are working together, right? So we can sort of, rather than just going in and trying to immunosuppress someone, that we can start to think about what are they doing? What's the goal of that? Can we then just go in and sort of maybe change the cells a little bit? Again, use something like a CRISPR so that we can modify how they respond so that they're not necessarily over-responding. But especially, this seems to be an issue in women. Understanding this, the role of inflammation in depression in women as something separate than in males. The other thing I would point to is that I have a paper out with Christina Dalla and Deborah Bangasser where we looked at CRF inhibitors, right? And so this is part of the HPA access. And one of the things that we've noticed is, because these failed miserably in clinical trials, they were great in animal models. And one of the things that we've noticed is that all of the clinical trials where they failed were in majority female populations. They were completely and utterly developed in male animals. They were never tested in females. There are absolute differences in CRF responses that Deborah Bangasser has shown. And the only clinical trial where it worked was all men. So here's something where you have a treatment that may really benefit men for depression, and yet it's not on the market because it didn't work in women.
Ooh, that's a good note to end on, because that's going to be a theme at the very end. I think I talked about it at the very beginning, right? That lack of translation. So thank you very much, Georgia. And it's my honor to now introduce Dr. Cecilia Flores from McGill University. I guess I can. I guess I could just add that in there just for some flair while I'm supposed to vamp while things there.
Thank you so much. It's a pleasure to be here. And congratulations and thank you, Lisa, for organizing this great event. And okay, so I'm pleased to be able to have the opportunity to share with you a little bit of the work that we do in my lab. So in this slide, I show results from another study showing once again that adolescence is a peak age of the onset for many mental disorders. And this is very important for many reasons, including prevention, because interventions at this age may have like the best outcomes. Also, there is a need to promote mental health during this age to achieve maximal benefits. But I'm very interested in trying to elucidate the underlying neurobiology of this heightened susceptibility in adolescence. And I think in this regard, the studies in neuroimaging in humans during normative development are shedding a lot of light, but also working in model organisms as the ones we do in my lab and in many other labs. So we know that adolescents are particularly vulnerable to certain environments, for example, to social adversity. For adolescents, the most important event in their lives is to socialize with their peers. And unfortunately, social adversity is common in adolescence, and influence has a negative impact on the mental health trajectories. So for rodents, also, socializing with other rodents peaks in adolescence. And if we do longitudinal studies in rodents, we can study how social adversity in adolescence can affect the brain and behavior in the short and long term. So in our lab, we have adapted the model of social defeat stress to adolescent males and female mice in a way that a big mouse attacks a small adolescent female or male mouse. And then after a few number of attacks, we house the animals in the same cage with a plexiglass divider that has holes, so the animals can still see and smell each other for the next 24 hours. And what we have found is that if we expose male adolescent mice to this social defeat stress, and then we let them grow into adulthood and we assess impulse control behavior using the go-no-go task, we found that those defeated mice remain impulsive in comparisons to control mice. But if we do this exact same experiment in adult mice, where we expose them to this social adversity and we wait the same number of days and test them in the go-no-go task, we find that they are not more impulsive, just indicating that the adolescent animals seem more vulnerable to this exposure. Another experience that is very common in adolescence is experimenting with drugs of abuse. Most people begin experimented with drugs of abuse in adolescence. And in the laboratory, we are interested in assessing the effects of exposures to drugs of abuse in the brain. And we use different drugs. In this case, I'm showing you the drug, the stimulant drugs, amphetamine. Although we administer this drug to the animals ourselves, we know that the regimen that we use has rewarding effects, and also that the drugs achieve peak plasma blood levels similar to the ones that are seen in humans with the recreational drug use. So in this experiment, I'm showing you data of male mice that are exposed to amphetamine in adolescence. Then again, we wait into adulthood, we run them in the go, no go task. And what we find is that the animals that receive amphetamine in adolescence are very impulsive, in contrast to the animals that receive saline in adolescence as they improve their impulse control behaviors across days. And again, this is a long time after the animals receive the drug. If we conduct this experiment in adulthood and we test them again in the go, no go task, waiting the same number of days as we waited in the adolescent experiments, we find no changes in impulse control behavior. And interestingly, if we perform the same experiment, but in female mice, we find in adolescent female mice, we give them with amphetamine, we find that in adulthood, these females do not show impulse control deficits. So going back to elucidating the neurobiology, we know that the prefrontal cortex is still developing in adolescence, in humans, and as well as in rodents. And we know by very beautiful work by other people that plasticity in adolescence seems to be open and closed by particular events, many of them happening in the prefrontal cortex. But I want to emphasize that not all adolescents are equally susceptible to experiences. There are significant individual differences, including those between males and females. And in my lab, we're trying to understand or characterize the processes that are involved in the developing adolescent prefrontal cortex, and to see how experiences that are common in adolescence impact these processes, with the hope of developing or characterizing molecular markers of vulnerability. And today, I'm gonna talk to you only about the first two parts that we do in the lab. So regarding the processes that are happening, this is a sagittal section of the mouse brain, and these are the dopaminergic neurons that send projections either to the nucleus accumbens or to the prefrontal cortex. It's been known now for over 30 years that the density of dopamine axons in the prefrontal cortex increases gradually across adolescence. And this is in comparison to the density of dopamine axons in the accumbens, which reaches adult-like density levels soon after being born, like 2 weeks in rats. And this gradual increase has been seen across species. But it was not until we performed an experiment in our lab in adolescent male mice, where we put 2 viruses, one at the level of the VTA, one at the level of the nucleus accumbens, in a way that dopaminergic neurons that would have reached the nucleus accumbens by adolescence will become fluorescent. And then we look at the brain of these animals in adulthood, and we wanted to see if they were fluorescent fibers in the prefrontal cortex, and they were. And of course, we run many controls, I can tell you about them, but we show for the first time that this gradual increase in the density of dopamine axons is because the axons are still growing from the nucleus accumbens all the way to the prefrontal cortex across adolescence. We were very excited about this finding because it's the first instance that is shown that long-distance axonal growth occurs so late in postnatal development. It's known to occur in embryonic or early postnatal life. Also because this is telling us that these axons remain very vulnerable to the changing environment. We also know that this protracted growth occurs in parallel to the well-known refinement in impulse control that occurs in adolescence. To understand how this phenomenon occurs. We have focused on guidance cues. One is called Netrin-1, which is a developmental guidance cues that organizes neuronal connectivity during embryonic and early life development by attracting or repelling growing axons. There are different receptors for Netrin-1. The one that I'm gonna talk about here is DCC, which is involved in the attractive function of Netrin-1. Netrin and DCC are important not only in axonal growth, but actually in telling the axons whether to stay or to continue growing to the intended target when they face intermediate points. And we have characterized very well the expression of these proteins in the dopaminergic system. I just want to tell you something important, that the dopamine axons that are in the nucleus accumbens and that remain in the nucleus accumbens have high levels of DCC receptors. But the ones that continue to grow to the prefrontal cortex, they do not express DCC or very little. So when we do the same experiment that I told you before, but in addition of turning these dopaminergic neurons fluorescent in adolescence, we also reduce their levels of DCC. What we find is that in adulthood, the number of axons that grow to the prefrontal cortex is significantly higher. Why? Because the axons have reduced levels of DCC and fail to recognize the nucleus accumbens as the final target, and instead grow ectopically to the prefrontal cortex. Something very interesting that we just found is that if we do this experiment with the two viruses to track the growth of dopamine axons to the cortex, we found that actually females have a significantly higher number of axons growing to the prefrontal cortex in adolescence. Now, we don't know why this is the case. I think, and I can tell you later why, but I really think it's because we're putting the two viruses, so we're doing the axonal tracking at the same chronological age between males and females. So I think that the processes of axonal targeting recognition in the nucleus accumbens is advanced in females than in males. Another interesting difference that I wanted to tell you about today is that if we take adult male and female mice, and we look at the dopamine transporter, so at characteristics of the dopamine axons that are in the cortex, we find that the females have more axons expressing the dopamine transporter than males. This, to me, seems very important because the dopamine transporter is the main way in which dopamine neurons take dopamine back from the synapses. It's also the major target of stimulant drugs of abuse. So just very quick summary. So dopamine axons continue to grow to the cortex. There seem to be sex differences, perhaps in the timing, and the molecules that mediate desegregation between mesolimbic and mesocortical dopamine pathways is the DCC receptors. So what about the experiences? So we study different experiences in our lab, as I show you, drugs of abuse and social stress, as many other groups. And what we have found is that in males, if we treat male mice with the amphetamine regimen that I told you about it before, but then 1 week later, we look at the levels of DCC when we think that the axons are undergoing these targeted recognition events, we actually find a reduction in DCC expression, and we have replicated this many, many times. So this is telling us that perhaps the axons are making targeting recognitions and are growing ectopically to the prefrontal cortex. And indeed, we did the same experiment with the two viruses, but in male mice that receive amphetamine in adolescence. And then in adulthood, whoops, I don't know why it's not, okay. Okay, so in adulthood, what we find is that here, I think it's just taking a bit. Here you can see the animals that receive saline. These I show you now, the fluorescent fibers. But the white arrows indicates here in the animals that receive amphetamine in adolescence, how many axons grew to the prefrontal cortex in these animals, showing that the axons that should have remained in the nucleus accumbens are now in the prefrontal cortex. We have quantified this with using stereological methods, so we see that it's across different regions of the prefrontal cortex, but we also, I think, okay, something happened here, but doesn't matter. But we, sorry, okay. So what we find is that the axons that are now in the animals that receive amphetamine in adolescence have more dopamine transported, resemble the female phenotype that I told you before. If we overexpress DCC receptors in adolescence, we prevent the effects of amphetamine in male mice. We don't see any of the changes. This is done with CRISPR in collaboration with Jeremy Day. And we don't see also the changes in impulse control. And this resembles to us the female-like phenotype that I told you at the beginning. And in fact, if we treat females with amphetamine in adolescence, we don't see this dysregulation in DCC, and we don't find the changes in dopamine connectivity and function. So just to summarize, in males, recreational like amphetamine triggers ectopic growth of mesolimbic dopamine axons to the cortex. The axons seem to retain the properties of the intended target because the dopamine transporter is mainly expressed in the nucleus accumbens, but not in the prefrontal cortex. This associates with impulse control deficits in adulthood and is mediated by DCC receptors and can be prevented, and females seem to be protected against these effects. So what about the social defeat stress? Well, we're using the model that I told you, but something that I didn't tell you is that 24 hours after the last defeat session, we test the animals in an interaction test where we find that the animals that are defeated show social impairment, but not all of them. Some mice, in spite of having been defeated or bullied by these mice, they show a resilient phenotype to this social avoidance. And what we did is, what happens if we expose these animals to the social defeat? Are they, does this experience induce targeting errors? In what we find, but remember that we can segregate them by their social behavior in adolescence. So what we find is very interesting. These are males. These are the adult mice showing in white, the white arrows, the dopamine axons that continue to grow to the prefrontal cortex. And you can see that in defeated mice, there are more axons that grew ectopically to the cortex, even though these animals did not show a social phenotype. They were not affected socially in adolescence. So that is just a cartoon to show. But here you can see that the animals that did show a social phenotype in adolescence, actually they do not show this ectopic growth. They look like controls, indicating that perhaps this process is modulated by how the animals respond to the experience early in adolescence. In females, what we find here, I show you again that the females have increased growth of dopamine axons growing to the cortex. But what we find is that regardless of the social phenotype in adolescence, females that are exposed to social adversity in adolescence actually show less dopamine axons growing to the prefrontal cortex. And we quantify all this with the stereology. What about the mechanisms? I can tell you very briefly that the Net1 system, the DCC receptor, seems to be involved in the effects in males with different constellation of changes in resilient and susceptible mice. But in females, we have not seen any changes in this guidance cue, so other processes are involved. I told you about the impulsivity that is seen in animals that are exposed to social adversity in adolescence. In females, what we see is that if we do the same experiment and then we let them grow into adulthood and do the go-no-go task, And this time we do hierarchical cluster analysis and segregate them. We find that there are good performers and bad performers. We find that, interestingly, those females that show social protection in adolescence are the only ones that show cognitive deficits in adulthood. And this seems to be like a trade-off between preserving social behavior in adolescence, but then developing cognitive dysfunction in adulthood. And without going into detail, I think there is beautiful work done in humans showing this trade-off for resilience in adolescence that I can tell you about. And just to summarize, so social stress induces social deficits in both sexes, but this doesn't result necessarily in ectopic growth and doesn't protect against cognitive deficits. In males, there is ectopic growth that seems to be involving the Netrin-1 system, but in female, the Netrin-1 pathways is not involved and the opposite phenotype is observed. And I just want to think that this division of resilience versus susceptibility classification is problematic because it's domain specific, vary according to chronological age, and is sex specific. And I just want to share some thoughts that we think that alterations in dopamine axon targeting and growth in adolescence induced by negative or perhaps positive experiences may be a contributor to sex-specific mental health trajectories. And this is just to thank my group, and thank you very much for your attention.
We have time for 2 questions. What I found particularly interesting is that, the resilience versus susceptibility, and of course, the sex differences that you see, and also in terms of mechanism. But I think it's a really important point that resiliency, we think, oh, you're resilient, it's all fine, and there's no sort of long-term neural or molecular signature with resiliency, but your work doesn't suggest that that's true, correct?
Yes, exactly that.
Yeah.
Yes.
And I think the same thing with was shown in with chronic restraint stress, like 3 weeks of chronic restraint stress. If you wait, that causes a signature in the hippocampus. And if you wait 1 week later, then it doesn't, it goes away. And so the sort of long-term effects are gone. But actually, if you look molecularly, they're very different. And there's a sex difference in that as well. So I think that's just underscores why sex is so important. Any questions while I was rambling? No? Okay, thank you so much. Okay, now we went human, animal. Now we're going back to human. I assume, I shouldn't make assumptions, should I? Mostly. And up next is CAMH's own Dr. Meng-Chuan Lai. And he's going to talk about integrating sex and gender in autism.
Great. Thank you so much, Lisa. And thank you everyone for staying here till the end of the session. So I'm going to talk about this condition called autism and how we can integrate sex and gender angles into understanding of autism. As you might know, and Megan has given us brilliant introduction to what this condition means. It's a neurodevelopmental disability condition, but part of neurodivergence as well, like existing across the population. It occurs in many people. Don Triplett is the first kid who was described as being autistic in 1943 in the English literature by Leo Connor, and who passed away, I think, last year at the age of over 90. We also have Temple Grandin, who has been a very successful animal science researcher, who's an advocate. We also have in our country, Carly Fleischman, who is a host of a TV show, but who's also non-speaking and using technology for communication. and we have Stephen Witcher, who is an autistic man who has savant skills. So you can see that there are a range of people who are diagnosed with this condition or who are autistic. And how is that related to sex and gender? And Megan cited this number yesterday, that if you read textbooks in psychiatry, clinical medicine, psychology, you often get this number of 4 to 5 to 1 male to female ratio. And this meta-analysis summarizes, if you stratify those epidemiological studies by how they ascertain individuals to be autistic, you see that if you're based on current educational or clinical database, you get a higher ratio. But if you look at the studies that went into the population and conduct an epidemiological survey using the instrument that we're having right now, you have a lower ratio. And this might be flawed, as Megan pointed out yesterday, that the instrument themselves might be flawed. So we don't know if this is accurate ratio. But there's a discrepancy here. So there are two messages here. One, the current practice missed some autistic girls and women, or assigned females at birth, actually. But there might still be a sex differential probability of whether one become autistic or not. A few years back, I was invited to write a popular science article and did a scoping to look at what does the science say about this, right? And then when I look into the neuroscientific literature, you can see that across different modalities, you get the ratio about all the way from 15 to 1 to 9 to 1 to 6 to 1, or the latest like 5 to 1. So essentially, not only the general population rates are biased towards assigned males, the literature are actually more exaggerated on that. So this is not, too different from any other conditions we're talking about today. I mean, we have a substantial underrepresentation of the data and the population for us to understand this condition. And the problem is actually more than that, because many of those studies in humans have only one index, which is a sex/gender, and sometimes they do not really make a clear distinction between sex and gender at all. So you have a label, a binary label, by the way, and that's actually very flawed in a way that I'm going to share with you. All the data later on will be contingent by that. And despite we know there are many mechanism-related factors in each domain that's important, current literature really does not tell us much about how they play out in understanding autism and neurodevelopmental conditions. However, I think it's important to have a map to think about how this may be helpful for us moving forward. So this basically is the slide to answer the title, which is how we integrate sex and gender into autism understanding. And I propose that there are multiple ways, actually quite complex ways. So first of all, it can be related to how the mechanisms associated with sex and gender relates to the emergence of autism as a neurodevelopmental condition. It could be some moderation effect, like attenuating certain factors. It could even be a mediator, like the factors or mechanisms associated with sex differentiation, for example, could play some roles in the emergence of autism. Then the phenotypic presentation, not only behavior, but also the brain, could also be moderated quite a lot by sex and gender. So how would the phenotypes, behavior or neural, be modified by one sex or gender? And that's related to recognition and diagnosis bias. And this understanding could even come back to help us rethink about how the definition could move forward in DSM-6 and ICD-12. And then, based on these understanding, hopefully we can have some support strategies. So I'm going to start with the recognition part. So this is a study that I was very lucky to be involved, led by Alana Woodcock in University College London in Will Mandy's group. And this is really the only study that I know so far that took an experimental approach to understanding potential gender bias. So at the time, we created some vignettes, and I sort of regret that we named it that way, but anyways, I'll come back to that later. So it's a 2x2 factorial design. We created some vignettes describing kids on the spectrums and behavior features and presenting them to elementary school, primary school teachers in the UK and asking them how likely you will refer this kid for autism assessment. It's a 2x2 design. So we have this behavior description profile that some of them are more subtle, and we call it female phenotype, which is actually not correct, but more subtle phenotype versus more classical phenotype. So for example, he tries to join in with other children but tends to be ignored. That's more classical. Or best friends with one girl in the class, although does not seem to be friends with any other children, so more subtle presentation. But the other manipulation was that there is a name attached to the child that implied the gender perceived by others. So what we found was that there's a significant interaction. When the presentations were more classical, the teachers didn't really make a huge differences in terms of the likelihood they will refer them for assessment, no matter they are perceived as a boy versus girl. But if the presentations are more subtle, and also if the name of the child implied the child would be a girl, then it's less likely the teacher will refer the child for assessment. So, and I think this is a quote that we got from one of our participants a few years back, which I think perfectly illustrate what the problem is. So moving on to the phenotypic presentation side of stories, and I think Megan alluded to many features that could imply the so-called nuanced features. And I'm going to give you a few examples about what might these nuanced features be like, and why might them not be able to be picked up by the current diagnostic practice if you're not paying attention to these. So this is a great study from my good friend Claire Harrop in North Carolina, who did an eye tracking study, and using the time of the child looking at the faces of the protagonist in the video as a proxy of social attention. So Claire recruited 4 groups of individuals, autistic and non-autistic school-age boys and girls, and then show them these videos. And as you can see in this socially rich scenario, there is a kind of like cascade relationships. So non-autistic girls pay the most attention to the faces, followed by non-autistic boys, followed by autistic girls, and then followed by autistic boys. So there's a main effect of autism diagnosis, but there's also a main effect of sex/gender. At this time, it's not distinguished. And so autistic girls lie somewhere in between in terms of the attention to social stimulation, approximated by attention to faces. And we sort of like replicated this in the European data set aged from 6 to 30, led by Teresa Del Bianco in the UAIMS data set. And literally, it's the same finding that if you presented them with dynamic videos, then there's a cascade effect showing exactly the same pattern. So if you look at social attention, then autistic girls are somewhere in between the non-autistic part and also autistic boys. Another phenomenon Megan talked about yesterday is this idea of camouflaging and masking. And I think one of the earliest illustration of this is the schoolyard playground behavior coding. So this is by Michelle Dean in Connie Kessler's group in UCLA. And what they did is that they went into schoolyards, playground during recess, and observed the behaviors of the children, and then coded how they interact with each other. And there are 4 groups. Non-autistic boys, non-autistic girls, autistic boys, autistic girls. Not surprisingly, unfortunately, autistic boys and girls spend most of the time in solitary activities, so being alone. However, autistic girls spend quite a bit of time in what they call joint engagement activity. So being aside with other children, not exactly in a structured play, but not exactly alone either. So if you look from afar, you might think, okay, this child is engaging with people, but if you don't know what exactly that relationship looks like, you might miss them being autistic. So this idea is that this masking idea is not only a behavior of the person, but it could also be how they are perceived or they are protected in the environment. Another very good example I would like to allude to is this experimental design by Henry Woodani in Southampton, I believe. So they conducted this interactive drawing test, which is essentially Winnicott squiggle test. So they invited autistic boys and girls and non-autistic boys and girls, and the autistic boys and girls are matched in their autism presentations based on parent report, and they're matched on their theory of mind test. But if you look at the frequency of interactions during this drawing test, autistic boys are interacted lower compared to non-autistic boys, but autistic girls actually interact the same level compared to non-autistic girls. So despite having similar levels of standardized measures from parent observations, I mean, they act a bit differently. So with Laura Hall at the time in University College London, so we created measurements from self-reports based on qualitative studies of adults' experiences of masking and compensating. And this is now widely used as camouflaging autistic traits questionnaire. And William I in our lab is now moving this towards other populations and more theorization as well. So I want to mention this thing as well. I mean, this is the work with Jill a couple of years back, and we did a scoping understanding what are the health presentations in autistic boys and girls as well. And autistic girls tend to have more issues in general compared to autistic boys, but non-autistic girls as well, and also higher prevalence of endocrine reproductive issues, for example, PCOS. So there are things that needs to be paid attention to. What about the brain? So the idea of sex moderating the brain changes could be illustrated quite well by these two kind of presentations. So if you think about the left-hand side, if the brain areas associated with autism are the same across males and females are less likely to be diagnosed autistic, so there's some quote-unquote protection happening, they need to be having more changes in the brain compared to boys. So that's what we call quantitative sex moderation. But it could also be the case that the brain areas are very different in the two sexes, and they emerge, so involving different brain regions. So with Chris Hummel using the PON data set led by Abdul Kiev and Gustu and Jason Lurch, we essentially tested this idea of whether there is increased changes in autistic girls in the brain in the same area compared to non-autistic girls than autistic boys compared to non-autistic boys, and we didn't really find any evidence to that. On the other hand, we actually find the brain areas involving the two sexes are quite different with each other. And the latest development on that is led by Sashi Bedford in Cambridge, UK, in Richard Betham's group. And this is really one of the largest studies that we have and most sensitive because we're looking at brain changes using this brain charting mechanism, which is basically a growth chart of the brain structural changes developed by Richard. And then with the amalgamation of many open access and available data sets across Europe, North America, mostly white still, like it's not really diverse population, but that's the largest we can get. And this is really what we know about autism in females in the past 10 years. Like this is the sample size we've gotten in the past 10 years. What we found was that if you look at regions specifically showing a sex-dependent pattern of autism, non-autism differences, we do have this volume differences at the temporal regions that has been shown to be quite important to autism that showed a sex differential relationship. But if you conduct a sex stratification analysis, what we found is that it's interesting to show that if you look at different brain metrics in the autistic groups, so the cortical thickness changes in males versus in females are not too dissimilar. They're actually quite significantly similar to each other. But the cortical volume and surface area, especially surface area, if you look at the coloring, they're actually quite different. So the autism-related features in the brain are quite sex-dependent, especially in the case of surface area. That does not occur in the ADHD cohort that we have. So talking about emergence a little bit, how can we leverage brain imaging to understand any implications about emergence? And this idea comes from if the likelihood of one being autistic is somewhat related to sex differentiation mechanism, and if brain is a readout of sex differentiation mechanisms as well as autism mechanisms, then if we can generate a model predicts the sex label of the brain based on T1 weighted image, that model might predict less well for autistic females because the two mechanisms actually go across each other. It might work better for autistic males because they're actually on the same line. So this is led by Cecilia Flores and Han Peng. I'm very glad to be part of that collaboration. Using the UK Biobank to generate a deep learning model predicting sex label with great accuracy, and then using that sex prediction label in the autistic cohort across two cohorts actually, and essentially showing that if we're having a prediction label lower than 0, that means the prediction is worse in the former group than the latter group. So essentially, the three hypotheses were confirmed that the sex label prediction model worked worse for autistic females than autistic males. It worked worse for autistic females than non-autistic females. It works better for autistic males than non-autistic males. It works better, the discrepancy is clearer in the younger cohort than the older cohort. So there's something happening early in life. It doesn't seem to be the case in ADHD cohort. However, this is not implying the misconception of autistic people have an extreme male brain, because what happens here is that if you look at the individual prediction distribution, most female assigned at birth are predicted as females. It's just the distribution differences, like results in the less well performance of the sex prediction in the autistic females. So that suggests there's some convergence between the two axes. And then I would like to refer to some animal work. And before that, this is work from Donna Whirling in Wisconsin, essentially showing that there is some convergence at the molecular level. So there are some upregulated genetic modules in autistic postmortem brains. And note, these are still mostly autistic male brains, so we don't know much about females. They are involving the immune glial cell axis. And Donna basically suggested that if you want to look at the convergence of the two, you have to look at how they are related to sex differentially expressed genes. And what was shown was that those autism upregulated modules are also mostly those who are more expressed in male than female, especially in prenatal periods of time. And that brings us back to Margarzi's model here of if you think about the rodent sex differences in the brain, it's not only the size differences, but also the microglia activation pattern. And that is a, for sure, hormonal related, but immune mediated model as well. So the idea was that if there's this likelihood of male individuals assigned at birth being more likely to be diagnosed with autism at some point, the biological mechanism could be associated with the inflammatory immune mechanism side of, or the tone of that. So finally, I want to bring us back to how does this mean for us clinically? I mean, there's still a lot of work to be done. So a few years back, my good friend Xiaoyan Lin and Stephanie Amos and I wrote a kind of a memo to clinicians about, okay, based on what we know about the nuanced phenotype, how can this be applied to the clinical interview or clinical diagnostic process? And this is really based on the DSM criteria. And we're basically saying that, okay, although this is the language used in the criteria, there are some interpretation that you can pay attention to help us capture those nuances. For example, the example Megan gave yesterday about learned eye contact and how that can be recognized properly, rather than just the absence versus presence of eye contact. I think that's one of the key important things. And this has actually been incorporated. It's not from our writing, but like the DSM-5 has a text revision last year, and they basically had incorporated what happened in the past 10 years into their text revision, not the criteria per se. So if you look at these ones, this understanding of compensation, that was there already 10 years ago in 2013, the initial version. But last year, they added this whole bunch of description, which is essentially reflecting what we talked about, the efforts into understanding or navigating social situations and how that can be laborious. The intersection to gender, I didn't really mention too much, but it's just too little. I mean, there is increased co-occurrence of gender diversity and autism. Lorna Bo, a medical student in Cambridge, and Anna van der Meesen, womenmind funded postdoctoral fellow, we did a scoping review of how does the transgender care guidelines say about considerations of autism at all. Not too much. Only 9 out of 22 that actually talked about this. But also they talked about a range of important clinically relevant topics. For example, autism shouldn't be a reason to preclude one from receiving gender affirming care. However, the assessment process definitely needs to consider the needs of autistic individuals properly. And we're doing a Delphi study to start to understand how does autism, how can we properly understand autism and diagnose autism in gender diverse individuals, led by Dr. Kate Cooper, UCL. So finally, I just want to bring your attention to this one. I think we've made some progress. So this is an expert panel report with a group of autistic women, parents of autistic females assigned at birth, and scientists that recently published in Lancet Child Adolescent Health, essentially highlighting what we know about and what we didn't know and what we should pay attention to more in the future about autism in assigned female at birth, considering the past and also future clinical directions. I would like to end by this paragraph from my student Kelly Moo essentially outlined what the problem. If there are people excluded from our initial research, and they are further excluded because they have co-occurring intellectual disability or other problems, then you have a bias sample, and in this case, male. And then this biosample being applied to our practice and then reproducing knowledge that's further biased. I think we need to fix that problem. Thank you all, and thanks my lab member and founders.
Thank you so much, Dr. Lai. That was, again, a masterclass. I think a lot of your work highlights when the diagnosis is sort of centered on the male standard, we can lose people, and that's a real problem, and also was highlighted by Megan's talk yesterday. Are there any questions for Dr. Lai?
Hi, I probably just zoomed out for a second or so, but could you just let me know what's your opinion of the male brain theory in relation to autism?
Yeah, so the initial extreme male brain theory by Simon Barrington, who's actually my PhD advisor, is about cognitive level differences. And there was a presumption that there is on average sex differences on certain domains. In his presumption, the differences between systemizing, so folk physics versus emphasizing folk psychology. And he presumed that there is this emphasizing systematic differences on average in the typical sex, in the typical population. And then autistic individuals actually showed a pattern that's extreme to that. And at that point, he didn't really differentiate whether there's differences in autistic males versus females. So it's a cognitive level theory, and he proposed that this cognitive profile may be related to prenatal mechanisms, for example, sex differentiation mechanisms. And this was taken very far from the community to say, okay, autistic females has to look like autistic males, or they need to be more extreme in a lot of cases, which is not exactly what the theory proposed. I would argue that theory was perceived in a way unintendedly that's led to a lot of inequity for people who are on the spectrum being diagnosed. I think the better way to understand that theory is about that convergence idea. So it's actually proposing whether there could be mechanisms from sex differentiation and from autism that's actually overlapping with each other. So you should actually take a look at that in a sex stratified way in terms of its prediction. So you look at what it looks like in autistic females versus autistic males. It's a cognitive level theory. It doesn't really speak to the other domains of the brain or the physical domains.
Yeah, I understand that, but I'm an obstetrician. Also, I'm also a gynecologist. So of course we worry about what's happening in the fetus during the pregnancy. So do I need to worry about the testosterone levels of the female fetuses, or what do you say?
I don't think that evidence there is sufficient to say this, because even the prenatal predictions, for example, the prenatal testosterone or prenatal estrogen data that's from the Danish biobank showed that it's never close to individual prediction accuracy at all. It's like it's a group level differences, and not testosterone actually. Both testosterone and the delta-4 pathway and estrogens are all elevated at the group level for those who are diagnosed autistic versus not later on. So I think it's at most a marker for some process that's happening, not only hormonal, but maybe other immune process as well. And we even don't know how that interacts with the genetic background of the person, right? So none of these environmental factors, except valproic acid, I think that's the only one we know quite well, that could be related to later neurodevelopmental outcomes. But I don't think we have any data to suggest on the hormonal side of things yet.
Thank you very much.
What about congenital adrenal hyperplasia? Do you know congenital CAH?
Yeah.
Is there, I was just thinking about that in relation to Inger's question. Is there any evidence that there's a higher prevalence with?
Yeah, there are some studies in the CAH cohort showing that the rates of autism diagnosis is higher in this population, but also autistic traits are higher in this population. Though the issue there is that it is related to hormonal level, but there are other things happening at the same time. So you just don't know the confounding factors and so on.
Yeah, I have a bit of a bias on the hormone part just because I love hormones. I mean, I did take up a question. We got coffee, but if is it quick? So I feel bad because I took up time.
You showed the sex differences, and I think was it brain mass or cortical mass? I just wondered, do we know anything about what that means clinically?
I don't think so. I don't think so. Yeah, brain imaging study has this limitation. It's not too far from phrenology, right? It's size differences, and some biology, associated with different metrics like cortical thickness versus surface area on the neurobiology of that. Though it's cross-sectional data, it's cutting across lots of age groups. We don't know the developmental trends of that. So, yeah, I mean, I think we need more cross-discipline integration to get to what it means clinically.
Thank you so much, Dr. Lai.